All right, so I'll start with a little introduction. So my background is primarily in bioinformatics and the analysis of gene expression patterns. So I'll do my best to give a little bit of an introduction in the data generation, which is really mostly Greg and Mimi's forte. And then I'll sort of transition into some of the data analysis that we've done on the initial data that we've generated. So gene expression changes across space and time are really important markers and important drivers of craniofacial suture development. And classical examples of that are twist and FGR2. So twist is primarily expressed in the suture mesenchyme, whereas FGR2 is expressed primarily in the osteogenic front. And functional disruption of either of these two genes leads to premature suture fusion of the coronal suture in both human and in the mouse models of these genes. So knowing these sort of patterns of differential expression across space and time for craniofacial sutures is very important, but a comprehensive profiling of these sutures hasn't been done. And this is something that we are trying to rectify in our project. So what we are doing is using laser capture microdissection to dissect both the osteogenic front and the suture mesenchyme from up to 11 major craniofacial sutures. And an example of that is illustrated here for the interfrontal suture, where we have dissected both the osteogenic front and the the suture mesenchyme shown here. And we are doing this for 11 major facial sutures, which differ in their orientation. So we expect to see most of the gene expression differences. So most of the gene expression profiles to be fairly similar across sutures, but there are also differences between sutures in the way that they are overlapping. So some of the sutures are end-to-end, some of the sutures are overlapping. And there are also differences between homologous and non-homologous bone pairs that we're interested in profiling. So an overview of all the 11 sutures that we're going to profile is shown here, 11 in total, and these are mixed in location, so cranial and facial sutures, in their lineage, neural cluster-derived, mesoderm-derived, in their bone pair homology, so homologous and non-homologous bones, and also in their structures, as I mentioned, end-to-end or overlapping. And I've highlighted here with the asterisks the sutures that we've either already produced data for or that we are currently in the process of producing data for. And what I'll talk about today is mostly the interfrontal and the coronal suture data that we've generated so far. So for each of the sutures, we are going through the following data generation steps. So we dissect the sutures, we get the mesenchyme, we get the osteogenic front. We do 5 replicas, so 5 individual mice for each of these suture subregions. In the case of homologous bones, we pool the osteogenic fronts. In case of differences in non-homologous bones, we profile each of the osteogenic fronts individually. So we do RNA extractions for each of these subregions, and typically we get between 1 and 10 nanogram of RNA, which is not a lot of material, but we get good quality, so RINs of 7 to 8. Because of the amount of material that we're generating, we have to do SPI amplification. And during this amplification, we are basically generating total RNA amplification. But this is done in such a way that we suppress rRNA transcripts. So we can do total RNA profiling without having a lot of reads going into rRNAs. So we do total RNA sequencing, Illumina high-seq, RNA-depleted material, and we're generating paired-end 100 nucleotide reads up to a target depth of about 40 million read pairs. That's what we're aiming for each of the suture regions. So the graphical overview here shows some of the data type that we're going to generate. So for the wild type sutures, we focus mostly on E16.5 and E18.5 development stages. As I mentioned, we pool in the cases of sutures between homologous bones, we pool the osteogenic front, so we generate 2 data types. And in cases of non-homologous bones, we generate 3 different data types, so the 2 osteogenic fronts and the suture mesenchyme. Wild type 16.5 and E18.5, except for the coronal suture, which we also profile at E14.5 because it's one of the earlier sutures that develops, and also because we are interested in studying it in the mutant mouse models. So for wild type, we are going to be generating about 285 profiles. And then we're also going to profile the different mutant mice, which are shown here. So the FGR2 mouse and the twist mouse. For APERT, we're going to generate all the E16.5 and E18.5 profiles. Basically a similar expression profiling overview as the wild-type mice. And for the twist mouse, we additionally do the interfrontal and the coronal suture at E16.5, E18.5, and for the coronal also at E14.5. So that gives us an additional 350 mutant profiles for 635 profiles across the entire project. So some of the steps in the data processing. So we start out with the raw reads, and I've underlined here the data types that we are submitting to the hub and that are going to be available through the FaceBase website, and for the coronal suture they already are. So we start with the raw reads. We do assessments of base level qualities, adapt our quality filtering. We map the reads to the mouse genome, so the kernelized genome is MM10. We produce map-read BAM files and genome browser tracks, and we do also quality level control at the map-read level.
So we're not hearing you.
Oh.
So maybe like put the actual microphone higher.
Sorry. Is this better? No? No better?
I think you'll need to use.
All right. Is this better? Yeah, well, I'll go here. Hang on.
So turn it up. So it's green, red. Yeah. So do you want to use this or this?
I'll use this.
Okay.
All right. Is this better? All right. Yeah. So as I was going through, we do quality assessment at the raw read level, which is shown here. On top. Adapt our quality filtering. We map the reads to the mouse genome, so the current version is MM10. We produce map-read BAM files. At the map-read level, we also do quality control, so we generate a number of statistics so that we can check whether the data is consistently generated across all the different suture types. And finally, we generate gene, transcript, and exon-level read counts for each sample, and we make these data available through the FaceBase website. So here are just some general statistics of the initial data sets that we have been generating. So we get very consistent data quality, both in terms of the number of genes that we can detect expression for, the fraction of reads mapped across the data sets, and the origin of the read. So we detect typically about 16,000 genes per sample. We have about, on average, mapping rate of 80%. And we get sort of a distribution of origins of the reads that is very consistent with what we expect for total RNA-Seq data, with about half of it derived from exonic, another 40% coming from intronic regions, but we also get a lot of reads coming from intergenic regions, and these are interesting because we can actually start looking at non-coding gene expressions in these data sets as well. So here's just an example of the type of expression profiles that we're getting. This is the case of Ibsp1. This is a major structural component of the bone matrix, and we expect this to be highly expressed in the osteogenic front, but not as much in the mesenchyme. And this is what you see in both the coronal and the interfrontal suture data sets, where in coronal, both in E16.5 and in 18.5, we see it expressed in the osteogenic front of the parietal bone and the frontal bone. And very little expression in the suture mesenchyme. And the same thing is for the interfrontal, where we see it in the pooled sample of the osteogenic front, but not in the suture mesenchyme. So for the same samples, we're also looking at differential splicing. We're also looking at splice junctions, and those are sort of displayed in the tracks here above. So you can see exon skipping events, retained introns, and all the other types of events that you expect in differential splicing analysis. So of course, this is only one gene, and what we're really interested in is sort of profiling the expression differences between suture regions across developmental times, and also between our mutant and wild-type mice. So what I've shown here is the expression differences between the interfrontal suture regions. So we have on the left the osteogenic front, on the right we have the mesenchyme, 2 different development stages, and we have 5 replicates for each stage. There is about 624 genes that are primarily expressed in the suture mesenchyme compared to the osteogenic front, and another 700 or so genes that are primarily expressed in the front compared to the mesenchyme. And what you can also see here is that we get very nice reproducibility between the replicates, so that's always great to see. And we see these very clear, sort of distinct differential expression patterns as we're expecting. So looking at the genes that are primarily expressed in the osteogenic front, we see gene ontology categories that are sort of reminiscent of what we're expecting. So we see skeletal system development, biomineral formation, bone development, ossification and regulation of ossification. And we also see some interesting other processes such as sterile metabolic processes and cholesterol biosynthetic processes, which we can start to look into a little bit more as well. Of course, we are also interested in looking at developmental time differences, and we're also interested in looking at mutant versus mouse model differences. So here what I've shown is the differences that we detect across time for the interfrontal suture. So we have the suture mesenchyme on the left, the osteogenic front on the right, and these are the number of genes that are significantly differentially expressed for each of these suture subregions. So for the mesenchyme, we have about 531 genes that are primarily expressed at E18.5 compared to E16.5, and about 604 genes that show the reverse differential expression pattern. And what is interesting is that we see far more differential expression in the suture mesenchyme compared to the osteogenic front. And the genes that are differentially expressed in the osteogenic front also have a high degree of overlap with those expressed in the suture mesenchyme. So these may be more sort of indicative of general aging than anything else. And what we think this signifies is that most of the development, sort of most of the time development of these sutures is really driven by the suture mesenchyme. Now, what we are really interested in is starting to integrate the data across many different suture types. And there are many different ways that you can do this. This is one of them. Basically, classical hierarchical clustering analysis, where you're looking at the interfrontal suture on the left, the coronal suture on the right. We see the different suture subregions, so mesenchyme, osteogenic front, the time points, and the wild type versus twist mouse in this case. And what you can see in this very large differential expression pattern plot is we can detect differences in regions, we can detect differences in gene expression between sutures, we can detect stage differences, and we can detect differences in genotype. But what this figure also shows is that as you're starting to add these suture data sets, this type of analysis starts to break down very quickly because there are many different comparisons that you can make in these data, and there are many different gene lists that you can produce, and you start to eventually drown in gene lists that you're going to produce here. So what we really want to do is take a different approach and start taking the data as it is and try and see if we can integrate these data and learn what the pathways of development, of suture development are. So the approach that we're taking here is a new method that is developed by our collaborator, Bing Zhang, and it's called Multiscale Embedded Co-Expression Network Analysis. And the idea here is that rather than looking at the hierarchical clustering of your data, you're actually trying to build co-expression networks where genes that are related in expression during development, during time, or in the different sutures get sort of spatially located near to each other within the network. So we start out constructing these networks. So we take higher array data or RNA-Seq data, we build a co-expression matrix, and we build these sort of large networks where the genes are denoted by points, and the connecting lines denote significant correlation in their gene expression. Now, the problem is when you do this analysis, you start to build very large clusters. So you get these big hairballs that I'm sure everybody here has seen. So what you need to do in addition to building these clusters is actually start to identify substructures within these clusters. So what we are using for that is a multiscale cluster detection algorithm. So this is an unsupervised hierarchical divisive approach that can identify substructures within these larger gene expression networks. And this will help you to identify modular structures within your larger networks. And with these smaller expression modules, you can then start to interrogate them and see, okay, do we find modular structures within the larger network that are associated with differential expression across time, between suture regions? And because this denotes potential biologically relevant pathways, we can start to identify potential drivers and mechanisms of disease etiology. So what does it look like? So this is the interfrontal suture, a global gene expression network. This is obviously one of these big hairballs that is very difficult at first glance to actually analyze and actually find structure. But as you start looking at this network, you see that the different subregions of this network are colored differently according to their sort of internal density and internal organization. And if you start to zoom in on that, what you see is a modular structure begins to arise. So in this case, all these different submodules that are identified by the divisive clustering algorithm have different colors here. And then you can start to test each of these clusters for the kind of contrast that you're interested in analyzing. So we have begun to do that for the interfrontal and coronal suture data sets. So one of the modules that we identified here, based on the differential expression between suture mesenchyme and the osteogenic front, is indicated here. So each gene is a node in the cluster. Each connecting line is evidence for significant co-regulation across all our data sets. And then the genes are also colored according to their sort of importance in the network. And the genes that have red names are identified as significant hubs in the network, not just based on their connectivity in the cluster. So these genes tend to have higher connectivity in the cluster, but they also maintain this higher connectivity if we start perturbing these networks and we sort of run the clustering analysis with different parameters. So these are not only highly connected genes, they're also highly stably interconnected genes once we start perturbing these networks. And in this particular cluster, genes that are upregulated in the osteogenic front are highlighted in blue, and the ones that are upregulated in the mesenchyme are highlighted in red. And what you can see here, I circled two different clusters. So there's one here on the top left where we have SPP1 and FAX. So SPP1 encodes osteopontin, and FAX regulates osteopontin. So we have two hub genes that are important in bone mineralization, and we see many other genes in the same cluster that also are known to be involved in bone mineralization, including DMP1 and EMPP1. So there is a second cluster here to the right, which is enriched in sort of type 1 collagens, which are also important in bone formation. So we think with this network-based analysis, we can really identify interesting regulatory modules within our data that are, and this is not a diss on Go again, but that are sort of in a way agnostic to what is currently known and are actually trying to reconstruct modules out of the data that we have available rather than relying on Go and what is known. Just a quick overview. So this particular cluster is obviously enriched in many sort of relevant Go ontology categories. So collagen fibril organization, ossification, biomineral tissue development, regulation of ossification. So we do see that within this cluster, we find the things that we expect to see different between the mesenchyme and the osteogenic front. Here's another network now for the coronal suture. Again, comparing the osteogenic front versus the mesenchyme. We have a bigger network on this end, so the attention that I wanted to focus is on this little cluster here to the bottom right, which I have blown up here. So if you start looking at the genes that are in this little submodule within this larger module, we see PANX3, we see PTCH1, SP7, GLY1, TCF7, and all of these genes are known to be involved in sort of stem cell maintenance, stem cell regulation, differentiation of osteoblasts. So even within these large co-expression networks, we can also find modules that are relevant for sort of stem cell maintenance, and these may be important in sort of the development of the osteogenic front and the differentiation of the osteogenic front. Again, similar to the one that we found for the interfrontal suture, we see again highly relevant Go ontology enrichment, skeletal system development, ossification, and skeletal system morphogenesis as well. So the final cluster that I wanted to show is related to development time. So in this case, we compared E16.5 versus E18.5. Again, genes up at E16.5 are in blue, genes up at E18.5 are shown here in red. Now, this is obviously a much larger cluster. We have about 651 genes, so it's hard to make sense of this cluster a priori. But again, we do see sort of interesting candidates. So PANX3 is here again, this TGF-beta 3, and I think there's another SCUB2 is over there. So we do find interesting sort of regulators of development here, but obviously we need to start layering in additional data to start further subdividing this cluster into more functional modules. And as we are starting to generate more data for other sutures and other suture subregions, we can layer in these data to further break down these networks in interesting submodules, because the more data you start layering in and the more detail you have in your co-expression network, the better your network topology is going to be. Again, although this cluster is hard to interpret, it's still highly enriched for anything that you think would be relevant for sort of organism developmental processes, as I've shown here in this module. So with that, I come to the conclusions. So we have done our first analysis of the interfrontal and coronal sutures at different development times. So we will also compare wild type and twist mutant mice. And we've shown that with our LCM data, we can really sort of capture accurate and novel aspects of suture biology, and we can really get very nicely reproducible data of the different suture subregions. And I think we've also shown that by starting to do this co-expression network analysis, we can construct interesting regulatory networks and new candidates for hubs in these networks that could have regulatory potential. And with additional suture data sets and maybe also integrating some of the other RNA-Seq data sets that are generated by the other groups in FaceBase, we'll be able to further define these network modules. And another thing that I wanted to mention is that the general structure of a co-expression network can take many different data types. So you can start layering in protein-protein interaction data, and you can start sort of pulling in data from other sources as well to further define and to further build out these networks. And also to do, for example, between a twist and a wild type mice, you can look at network topologies and can see other disruptions in certain network modules that you can find that way. So with that, I just wanted to acknowledge everybody involved in the work. So Greg has done obviously most of the dissections and most of the RNA prep work. Mimi, Michael, so Bin Zhang is our collaborator. So he recently published a post-computational biology paper describing the method that I outlined here. And sort of our whole department is sort of specialized in this kind of co-expression network analysis. And then Stephen Potter from Cincinnati Children's as well. So with that, I'll take some questions.
Thank you, Harm. Okay, Phil, hold on.
Great talk. Just one question. You mentioned this just at the end, but on your expression sort of data, have you tried to overlay the protein-protein interactome?
So we haven't done that yet.
Because of course that's not going to be done in suture cells, but maybe you can still get some inklings from that.
Yeah, so we haven't done that yet, but it's definitely something that we're interested in pursuing.
So I guess I wanted probably more of a clarification. So I noticed that you proposed to start collecting samples for the coronal suture at E14.5, correct? And then the other sutures at a later time point. So at E14.5, the frontal bone and the parietal bone, they are really small, just the primordium, right? How well can you actually define the coronal suture at that point? And I guess probably a more important question is like when you look at your twist mutant, when do you begin to see gene expression changes at a particular embryonic stage that is really, you think is relevant to the premature effusion of the coronal suture?
So, yeah.
So the E14.5 is a challenge, and I mean, I've done a lot of this kind of dissection, and that's something we're sort of leaving for later and sort of trying to work out the best approach for that. It should involve some kind of staining, but when you stain, you have to make sure you still have good RNA quality. So working on an alkaline phosphatase stain will definitely show the primordia at that age. It's just a matter of keeping the RNA quality good. And then for twist, so I guess the earliest changes in the twist coronal suture is at E14.5. You see alkaline phosphatase can start to cross between the frontal and parietal bones, even at that early age. Although the synostosis of the suture itself is much sort of less efficient, I guess you could say, than in the 8-bit. So that's why we're going for the E14.5 in the twist as well, since it is affected at that early age.
I guess at that point, the region, the suture region, is really wide, right? How do you define that region as a suture based on some markers, or just how do you know where the suture is or where the osteogenic front is? Because that is still developing stage, right?
So, well, for E14.5, I would say like the frontal and the parietal are very close, which is again part of the reason why they're probably affected very early. So I don't think it's not too wide that you can't sort of claim it as it is like a pre-suture area. And like for some of these sutures, E16.5, it's also a fairly wide suture mesenchyme, and maybe you would say it's pre-sutural. But I think from what we're seeing even with the interfrontal, there's interesting expression there, and there's definitely expression differences between the osteogenic fronts and the mesenchyme that's in between them. By 18.5, it'll be more like a suture.
So, you may have noticed from my talk that I'm sort of obsessed with signaling, and particularly in the terms of the bone and suture formation. My understanding is that there's a lot of very interesting mechanical-based signaling that's going on back and forth in cartilage and bone formation. And I didn't see a whole lot of those types of things in your genes in your analysis. I was wondering if they weren't coming up as differentially expressed or if they were just buried in the networks there.
So they definitely, so our differential expression data is definitely more than what I've shown here in these networks. So I've just given some examples of submodules that we found, but there are additional modules that I haven't presented here today that probably have some of the differences that you're looking for.